Good day, class. Today's laboratory session will be about the extraction of genomic DNA from plants of medicinal value. The growing interest in traditional and alternative healthcare led to the increased attention that medicinal plants are receiving in terms of their potential as sources of bioactive compounds that can be used as treatment for certain medical conditions. This laboratory session is divided into two parts, part A and B. For part A, we will cover plant DNA extraction up to spectrophotometric analysis of DNA quantity and quality. Discussions covering part B will be made available next week, Thursday. We will kick off our discussion with background concepts prior to the laboratory proper. After discussing the methodology and presenting some expected results and data, we will discuss sources of errors as well as four sample problems, two of which we will be answering by the end of this session. Okay, so some preliminary concepts that we need to discuss. Of course, it's important for us to understand the nature of the molecule that we are dealing with, the DNA. So, a little about the biochemistry of DNA. DNA serves as the information carrying biomolecule for living organisms. Before engaging in any attempt to isolate DNA, one must understand how it is packaged at the cellular level. Prokaryotic and eukaryotic DNA are strikingly different in terms of their location, size, and structure. In terms of location, in contrary to prokaryotic DNA, which is found in the nucleoid region of the cytoplasm, eukaryotic DNA is primarily found in the nucleus. Other organelles, such as the mitochondria and the chloroplast, for the case of plant cells, also possess DNA inside them. In terms of size, eukaryotic DNA are large, ranging from megabase pairs to gigabase pairs, while prokaryotic DNA is usually small, reaching about megabase pairs in terms of their total size. Structure is also another difference between prokaryotic and eukaryotic DNA molecules. So in terms of this structure, I want you to refer to this figure in this slide. So the two illustrations actually demonstrate how eukaryotic DNA is packaged inside the cell. Key packaging levels of DNA that we will be discussing in the next slides that I want you to take note, please, are as follows: the nucleosomes, okay, the solenoid chromatin fiber, and of course the condensed chromosome, as you can see here. I want you to think of the eukaryotic DNA as a protein-bound nucleic acid material. So the proteins that actually bound to DNA molecule or to nucleotides are what we call histones. No? So histones are a set of alkaline proteins that help DNA condense further as it actually undergoes different levels of condensation. So in contrary, prokaryotic DNA is said to be naked as it does not have associated histone proteins. Okay. So here on the next slide, we are seeing, and we can actually appreciate here, the increasing levels of eukaryotic DNA condensation from the DNA double helix up to the metaphase chromosomes that are duplicated and that will actually have a double diameter compared to heterochromatin. So notice that there is increase in diameter size of these macromolecular levels of DNA packaging. So you can see from the DNA double helix with a diameter about 2 nanometers, you proceed with nucleosomes, structures that are um, said to be or are referred to as beads on a string, having diameters of 10 nanometer or 10 nanometers. Next, you have your chromatin fiber or your solenoid. It's actually your nucleosomes. They on a top view, you can appreciate them as a flattened or as a rosette-like, having a rosette-like arrangement. So when they're taken together, their zigzag pattern, you can see the chromatin fiber having 30 nanometers in terms of their diameter. Furthermore, as these chromatin fiber begin to develop domains called loop domains, the increase in the diameter is evident. You can see that it's already 300 nanometers. Heterochromatin is the last condensation level of DNA prior to the metaphase chromosome. And we can see that there's a doubling of the diameter size from heterochromatin to the metaphase chromosome. No? So this is a question, I think, that is provided in your pre-lab quiz. So I hope that you were able to get the correct answer. So, aside from the macromolecular packaging of DNA, it's also important to understand its molecular structure. When we talk about DNA in the undergraduate level, the first thing that comes into mind are the nitrogenous bases. We have the purines and the pyrimidines. No? So the purines, adenine and guanine, and the pyrimidines, cytosine and thymine. Purines are consisting of six-membered and five-membered nitrogen-containing rings. As you can see here, adenine, guanine are consisting of two nitrogen-containing rings. One is five-membered and the other is six. On the other hand, pyrimidines have only one six-membered nitrogen-containing ring, your thymine and cytosine. Aside from from these four nitrogenous bases that we have encountered so far, there are also other types of purines or other examples of purines and pyrimidines that you might encounter in your next courses or in your lecture. So aside from the purines adenine and guanine, we also have two other purines, hypoxanthine and xanthine. On the other hand, for pyrimidines, additional two pyrimidines that you will eventually encounter in your study of the biochemistry of the DNA are orotic acid and uracil, which is actually found in RNA. Now, okay. Together with our DNA backbone, the DNA backbone consisting of two important molecules, the sugar deoxyribose and the phosphate group that is linked to its carbon number 5, we can actually appreciate the spatial relationship of the nitrogenous base. So the nitrogenous base could be any of the purines and the pyrimidines that we have discussed in the previous slide. It's actually attached to carbon number 1 of our deoxyribose sugar. So together, we call a nitrogenous base with an associated sugar that is having an attached phosphate group as a nucleotide. So nucleotides, are the backbone or the um, fundamental units of nucleic acids. Okay, next. Yeah. Okay, so further examining the nucleotides as the building blocks of DNA, we encounter the phenomenon of specific base pairing among the four nitrogenous bases. So we have adenine pairing with thymine, guanine pairing with cytosine. This phenomenon is known as the Chargaff's rule, named after Erwin Chargaff, who observed that there are equal molar concentrations of adenine, thymine, as well as guanine and cytosine in most of the DNA examples or DNA samples or molecules that he examined across different living organisms. No? This specific base pairing of nitrogenous bases is actually due to the formation of hydrogen bonds between them. Let's take a closer look at those nitrogen bonds. So here you can see a diagram of thymine base pairing with adenine and cytosine base pairing with guanine. Okay. So the H bonds are represented by the dashed lines in between the base paired or the paired bases. No? So remember that a hydrogen bond is the attractive force between the hydrogen attached to an electronegative atom of one molecule and an electronegative atom of a different molecule. So usually the electronegative atom is an oxygen, a nitrogen, a fluorine, which all has or should have partial negative charge in this kind of association. So we can appreciate here the hydrogen bonds. No, also take note that adenine and thymine only has only the pair adenine and thymine will only have two hydrogen bonds, while the pair guanine and cytosine will have three hydrogen bonds. And actually, the number of hydrogen bonds, or I would say the composition of the DNA in terms of its guanine, cytosine, and adenine thymine percentage, or guanine, cytosine, and adenine thymine composition, will will have some implications to the temperature needed in terms of denaturing DNA. So you might encounter in literature, or definitely you will encounter in literature, that DNA with more guanine and cytosine are more strong, or is more strong in terms of denaturation temperature. It will require higher temperature for such DNA to actually separate. No? Okay. Now, let us take a look at how a series of nucleotides actually look in a double-stranded DNA. So, here we can see a diagram showing a stack of nucleotides, a linear stack of nucleotides, and a pair, or a strand that is opposite to it. So, aside from the spatial relationships between the DNA components, we can also appreciate in this diagram the anti-parallel nature of double-stranded DNA. So as you can see, one strand runs from 5 to 3 prime, and the opposite strand actually runs from 3 to 5. So the anti-parallel nature of DNA is described as when the head of one strand is always laid against the tail of the other strand, so you can see here. So remember that DNA, especially that of eukaryotes, must be packed at succeeding condensed levels. Therefore, it is inevitable for the DNA to maximize space. Imagine the double-stranded DNA to be forming a helix from which, when torsion is actually removed, so you can see here the double-stranded DNA forming a helix. If you remove the torsion from this molecule, the double-stranded DNA, for the case for the context of plants, or for the context of eukaryotic organisms, will look simply like a ladder, where the legs of the ladder are the sugar phosphate molecules, the backbone, while the rungs of the ladder represent the nitrogenous base compounds, or the nitrogenous bases that are bound by H bonds, no? By hydrogen bonds. We can summarize everything that we have discussed so far using the Watson and Crick's model of DNA molecular structure. So everything that we've mentioned so far actually fits to the molecular theory on the structure, or the theory on the molecular structure of DNA as proposed by Watson and Crick. Okay, so you can see the spatial relationships of the DNA components, the components of the nucleotides. Also, we can see that in order for the DNA to have a stable, or for DNA to, or in order for DNA to actually uh, ensure space saving, or to achieve space minimization, or space maximization rather, okay, we can see that double-stranded DNA in living organisms is actually of helical shape, no, or helical configuration, right? Now we proceed to the highlight of our laboratory activity. We want to isolate DNA from a living organism, in particular, medicinal plants that are recognized and endorsed by our Philippine Department of Health. It is important to take note of the key principles or ideas in DNA extraction. So one of the first things that I want you to remember is that the process of DNA extraction involves physical as well as chemical procedures that will lead to the shearing or breakage with of material with very high molecular weight DNA molecules. So we expect that DNA from eukaryotes that are usually very big in terms of size are covered or are within compartments of the cell, within the nucleus, within the mitochondria, within the chloroplast. So in order for us to access that DNA, we need to break those cell material. No? Okay, so this is a good picture. So for this activity, we will be using young leaves of known medicinal plants. From your pre-laboratory discussion notes, we have identified for you the 10 endorsed and acknowledged medicinal plants by the Philippine DOH. No? So for this activity, you will actually be using the young leaves of this known medicinal plants. So remember that within a leaf, within just a single leaf, you can find millions to billions of plant cells residing on just one planar surface of one leaf. So in each plant cell, you can see here the blow up picture, in each plant cell, we expect that DNA may come from the organelles the chloroplast, and even the nucleus. No? Okay, so the second key principle or idea that you want to be remembered or you want to remember from time to time is the fact that DNA, once extracted from your living system, from your cell or from a virus, is going to be purified or is needed to be purified by the removal of molecular components, mostly proteins. Because when you open the plant cell, you of course expect other components aside from nucleic acid, aside from DNA, or aside from DNA, you will expect another type of nucleic acid, the RNAs, and also you expect big molecules like proteins and carbohydrates as well as lipids. It's very important that contaminant removal must be done because if we're going to use our extracted DNA for other downstream processes, or let's say if we want to do in vitro manipulation of that DNA, it is important that the DNA is stable. And stability of the DNA molecule can only be achieved if it is pure. Okay? Now, the last and final idea or principle that must be observed in the process of DNA or in any protocol of DNA extraction is proper storage of the extracted and purified DNA. Usually, of course, in laboratory settings, DNA is commonly stored at low temperatures. So, yeah, 4 degrees Celsius to probably until negative 20 or negative 80 degrees Celsius to avoid enzymatic degradation and other unwanted chemical reactions. DNA is often stored as a precipitate, so it's allowed to precipitate or to be precipitating in the presence of ethanol at negative 80 degrees Celsius. It's very important that you keep DNA in that kind of preparation if you want to use it for long term, like 2 to 6 months. But if you're going to use DNA, your extracted DNA in within a week or a month, negative 20 or 4 degrees Celsius is actually a working or a temperature condition already. It's already okay. Now, so after extraction, analysis of quantity and quality of DNA must be carried out. For our laboratory activity, we will carry out spectrophotometric analysis of DNA. So in order for us to accurately determine the concentration of DNA in our sample, a UV spectrophotometer will be used. Now, the purity of a solution containing DNA, or our extracted plant DNA, can be determined using a comparison of the optical density values of the solution at various wavelengths. And to do that, or to start that, we will first discuss how we can quantify DNA using spectro data. So to determine the concentration of the stock DNA solution, we must consider two things. First, how much the DNA stock has been diluted to make the sample that we analyze in the cuvette. And second, of course, the output reading, the absorbance of the diluted sample that we analyze in the cuvette, in the spectrophotometer. Okay, so how do we do this? First, let's refresh ourselves with the concepts of dilution factor and absorbance. So the dilution factor represents the ratio of the total volume of the diluted DNA sample in your cuvette and the volume of the stock DNA solution that was used to make that sample. So it's always going to be the ratio between the volume of the diluted DNA sample in your cuvette and the volume of your stock DNA solution. So that's always your dilution factor. No? So since we're talking about volume, we expect that, or since we're talking about two values that are expected to have the same units, when you actually divide them, you expect that dilution factor will not have a unit. No, so the volume in microliters, as you can see in our example, will actually cancel each other out. Okay. All right. In terms of the absorbance of the diluted DNA sample, so you have done your dilution, or you've diluted your stock DNA. You counted, or you computed for the dilution factor using the parameters that I have mentioned, and you have subjected your diluted DNA sample in the cuvette under spectrophotometric analysis. No? So you of course expect readouts, no, absorbance readings. Okay, so in terms of the absorbance of the diluted DNA sample, we can actually simply refer to this table of accepted conversion values. No? So if we are seeking to determine the unknown concentration of our diluted DNA sample, which is what we really want to do, this information would be very helpful. Let's take a look further at another concept that will be helpful for us. No? Or how do we actually use this information? Let's take a look at the next slide. Okay, so here we can see the relationship between our the accepted conversion values on this slide and the absorbance value that we expect to be churned out by our machine, and the unknown concentration of the diluted DNA sample that we want to actually determine. So this is the major value that we want to calculate using our accepted conversion values. No? So let's say, for example, that we have this problem. You have a diluted DNA that read that produce an absorbance value of 0.09. So you want to know the concentration of your diluted DNA in the cuvette. Okay, so remember the formula for getting the unknown concentration. You just have to do transposition of the values. So that will be your the result will be the absorbance of your diluted DNA multiplied by the accepted conversion value of 50 micrograms per ml uh, for every one absorbance unit for 200, and using the wavelength 260 nanometers. Okay. So guys, I want you to remember that um, in this calculation, or in this particular problem, our assumption is that we are dealing with double-stranded DNA. If you will go back to the previous two slides, the table that I have shown you, okay, you have different absorbance readings under 260 nanometers, and that will and each absorbance reading is particular to the sample that you're analyzing, whether you're doing an analysis of single-stranded DNA or an RNA. Okay, so is this the exact concentration or the concentration that we are looking for? For the diluted DNA sample, it is. But since the question is framed as the sample being diluted, we expect that there is a stock solution. Okay, so the concentration for the concentration of the DNA in the stock solution will be obtained further. So how do we do that? Simply, we multiply the concentration of diluted DNA that we acquired, the one in the cuvette, with our dilution factor. So let's say, for example, we have a dilution factor of 100, right? The total concentration of DNA in our stock solution will be 450 micrograms per ml, following the input from from this slide. No? So you just have to multiply the dilution factor to your determined concentration of your diluted DNA so that you can get the concentration of DNA in your stock solution. Okay? Right. So now let's proceed to the next slide. Aside from the quantification of DNA, okay, quality check must also be done prior to downstream applications. So it can be done using a comparison still of the optical density values of the DNA sample at various wavelengths. Nucleic acids, the ones that we're dealing with, RNA and DNA, okay, depending on the composition of their bases, will absorb maximally at about 260 nanometers. On the other hand, proteins, which are the other molecules we expect to see in the cell and we expect to be present in our DNA extraction steps, and eventually we need to remove, they absorb maximally at 280 nanometers and 230 nanometers. Now, here, let's take a look. Um, if you will have a ratio, absorbance ratio, 260 280 absorbance ratio that is 1.8 or near 1.8, you can you can characterize your DNA isolate to be pure, to have high purity. On the other hand, if that ratio is above 1.8, it may indicate that your DNA sample is actually contaminated with RNA. Now, below 1.8, absorbance ratios that are below 1.8 actually indicate possible presence of protein or phenol. Alternatively, you can also check on the absorbance ratio between 230 and 260 nanometers. And if that ratio is greater than zero point 0.5, okay, that will suggest, or that actually suggests, that there is protein and or phenol contamination. So for DNA extraction protocols that involve these chemicals, and particularly phenol, it's very important to check whether your sample is free from it or not. Okay, so I'll show you a very useful table summarizing everything that I've mentioned so far. Okay, right, so here, just take note of the 260 280 ratios and 230 260 ratios. So the key value here is 1.8. So the more near, or the nearer your absorbance ratio to 1.8, the more pure your DNA sample is. Okay? Alright, so for the last part of our introductory concepts, we will be talking about medicinal plants. Now, in our country, in the Philippines, the following 10 medicinal plants are acknowledged and endorsed by the Department of Health for their proven health benefits. It was the intention of this laboratory activity for you to actually pick one of this medicinal plants and extract their DNA material. So we actually want to subject you to wet laboratory, no? In a more global scale, so I tried doing some very basic literature review in terms of statistics now. In a more global scale, attention is actually indeed pouring into research about medicinal plants. So I use the keywords medicinal plants, so taken as one keyword, and DNA. So using those two keywords, I went to check the repository of the National Center for Biotechnology Information, and NCBI returned an outstanding number of published literature and scientific articles. So you can see here from PubMed Central and PubMed, you can see more than a total of 15 or 4 or 16,000, now 16,000 scientific articles, publications that are associated to studying medicinal value of plants, and probably might have used DNA as an important unit of analysis. In fact, for this year alone, for 2020 alone, I checked Google Scholar and still use the same keywords. No? And what did I find out? There are about 2,290 relevant publications, scientific articles that are associated with the study of medicinal plants and their DNA, or possibly DNA as the unit of analysis or source of data. Indeed, this is actually a growing research area that perhaps, might attract your attention and focus when the time comes that you need to start doing your undergraduate research work. Going back local, our Department of Health has a dedicated unit for traditional and alternative health care, the PTAC, or the Philippine Institute of Traditional and Alternative Health Care. If we take a look at the databases of PTAC, we can see that it includes monographs and some advisories, some high-level data on medicinal plant research, and some books and reviews. No? In fact, one of the books that captured my attention is this one, the Encyclopedia of Common Medicinal Plants of the Philippines. This is a book edited by Fabian Dairit and colleagues, no? Two more colleagues. No? So, now that we have discussed all the background concepts, we will be proceeding with the discussion of our objectives and methodologies in the next part of our discussion.